So this dude, his name is William Smith. This guy is called the father of English stratigraphy.
English, huh?
Now, stratigraphy is basically, if you look at the rock layers, you will notice that they kind of look like layers on a cake. All right? Now, they very rarely look that neat, but that's the idea. All right? The idea is you're going to start finding fossils scattered through these layers. And so the idea is, as you look at them, logically, fossils in the same layer must be the same age. I know that seems really obvious to us, you know, like, duh. But again, understand there had to be a point at which the wheel was discovered, as obvious of an invention as that probably would have been. So some of these discoveries, you're kind of like, yeah, So as we look at this is the idea of stratigraphy. There are layers, and we're going to study this layering system that happens. Fossils in the same layer are the same age. Okay? Now, as you continue working your way through this, I could give you a whole lot more background if you really, really wanted, but I don't think you really, really want it. This next guy is a man by the name of Charles Lyell. Charles Lyell is going to be a massive influence on Charles Darwin. All right? In fact, what Charles Lyell is going to do to geology is going to strongly influence Darwin in the area of biology. So just kind of pay attention to that connection. Lyell is going to encourage Darwin to publish his work because there was another guy who was actually working on the same idea and just hadn't gotten it out yet. And so there's some fun little shenanigans that were played there. So Darwin was not the only one who thought of this. In fact, I think Darwin's grandfather had kind of formulated one of these ideas before. And so Charles Lyell, he puts out this idea of the principles of geology, and he came up with this, or at least popularized this concept called uniformitarianism. Okay? Let me explain, because there are actually two words that are very, very similar, but they have a slightly different meaning. All right? Do not confuse uniformitarianism with uniformity. Okay? So I'm writing uniformity on the board. Uniformitarianism is right there. How this works is this. In science, we believe that the natural laws have continued the same since the beginning, pretty much will always continue the same way. Those natural laws functioning the same way at all times and all places, that is the core basis for this idea of uniformity. If it worked back in the past, I'm assuming it'll work now. Like, that's the law of gravity. I don't assume is going to randomly shift. It works the same. That's uniformity. Uniformitarianism says the rate at which things happen, or what I'm seeing happen today, must be the exact same rate, basically the present is the key to the past. So if I see it happening today, I can extrapolate that backwards and make some assumptions about the past. All right? Now, we're not going to spend a whole lot of time arguing against this because there are a lot of places where this is going to help us. But what I want to show you is evolution is entirely based on this idea that the present and the way we see things happening today is the way they've always happened. Okay? So if we don't see cataclysmic floods, then there probably weren't cataclysmic floods. And if we don't see God getting involved now, then, well, evolution doesn't really have a place for a designer anyway. But you kind of get this idea that there is no stepping in the past and big things happening. And no, you're probably looking at just a progression of little steps over a long period of time. All right? And so he's going to push this idea of uniformitarianism. And what's going to start to happen here is it's going to start to deny a lot of the catastrophes. It's going to start to deny younger things and say that the Earth is probably older than we give it credit for. And it's going to start bumping the age of the Earth back, by thousands, then millions, and then, into the billions. And so this idea is going to start really pushing evolution forward, because Darwin's going to take a look at this and say, oh, well, if you have finch beaks, and they can all come from a common ancestor finch, then maybe all those came from a common ancestor bird, and maybe all those came from a common ancestor of something like that, and maybe all those came from a common ancestor, and maybe all those came from and then as you kind of progress back in time, that's why you need so many years, because it takes a ridiculously long time. All right, so Charles Lyell is going to do to geology what Darwin does to biology. Okay, now here's some assumptions. Like I said, this idea of uniformitarianism functions on this idea that the rates of things remain the same throughout a long period of time. Now, like I said, we're not going to spend a lot of time refuting this because there are times where this is true. There are times where the rates just kind of continue roughly the same. And if you don't have to appeal to a cataclysm, then don't. I mean, you don't have to quickly jump automatically to, well, there's a riverbed here. It must have been a flood that ripped through. No, it could have been just that water flowed for 20 years and slowly carved a riverbed. That could be the reality of it. But what they do is they apply this to things like the Grand Canyon, and they say, well, the Colorado River is carving it at so many a year. So therefore, if you think about how deep it is, it must have been carving this for so many insert number of years here. They refuse to admit that, no, actually, probably could be something that was carved by a massive amount of water ripping through that channel really fast. So let me show you an instance. In Mount St. Helens. So have any of you guys been to Mount St. Helens? Okay, Mount St. Helens used to be just an absolutely beautiful, mountain. I know, isn't it gorgeous? But what happened is, back in the 80s, they were watching, and I don't know, they either didn't realize it was a volcano, I can't remember the whole backstory to it, but as they started watching, they realized, something's happening to this mountain. So it started swelling and started getting all misshapen, and they realized, oh, this is a volcano and it's about to blow. And so I think they got a lot of people out of the area, but eventually it did, and Mount St. Helens, just lost its entire top. Okay, so Mount St. Helens just blew its stack. What's amazing is they said when a volcano like that erupted, there's actually a zone around it where you can't hear the eruption because it blows the sound waves upward. And so there was a section around the volcano that didn't know, I guess, that it had erupted. And then, obviously the stuff coming off the volcano killed everyone around. And it's kind of unfortunate. You're like, oh, junk. I think some people might have survived it, but I'm not positive. It's weird though. The fact that it might have been 60 miles around that you couldn't hear the volcano erupt. It was something weird. They explained it at the place, and it's been several years since I've been there. But Mount St. Helens is now more or less like that. I mean, it's pretty destroyed. When you go there, in the 80s it erupted, and a few years later it started the snow melt and the mud and all that after the eruption flowed down in the valley and just kind of covered the valley in layers of sediment. So you had volcanic ash, you had stuff that was blown away, you had mud. So you get these layers, which is kind of cool to be oh, we got an instant, strata layer laid down near Mount St. Helens really, really fast. I mean, within months, if not a year. And so you get this layer. Well, then as the snow started to melt, and you had a few other eruptions and other things, it washed down the side of the volcano, and it carved a canyon in that hardening mud that was 1/40th the scale of the Grand Canyon. So Mount St. Helens erupts, lays down layers and layers and layers of ash and mud and dirt and everything all around it. And then the water coming off of the snowmelt actually carves a really deep canyon. Like I said, that's 1/40th the scale of the Grand Canyon, and it carves it nearly instantly. I mean, we're talking, if not a matter of weeks, months. I mean, it's quick. And so we've got at Mount St. Helens, a really good empirical evidence of canyons don't take a long time to carve. In fact, sediments don't take a long time to lay down. Now, where this gets difficult is because part of the reason they discovered some of these things with the rock layers is because you can see the same rock layer going across a continent. Like you can see this, so it's not like Mount St. Helens where it's just in this area around the volcano. It's like, no, could you imagine a rock layer that went across all of California? Well, then you have to ask yourself, that was a really big explosion, or that was all buried under the same layer, or, you know, so that's where you bring up some of those more difficult questions that you would then have to answer. Now, let me show you a video on this really quick, just to kind of show you how that would work. And there it is. Okay, so this is a video about Mount St. Helens. There are obviously better videos on the Grand Canyon out there, but that is the basics of it. As you compare the Grand Canyon to Mount St. Helens, you do see a very similar canyon structure. Again, we know that Mount St. Helens formed rapidly. Now we're going to come back to Mount St. Helens because when it erupted in the 80s, it blew its top off. And then one of the cool things that started happening is it started forming a new lava dome. And so the lava and the magma that's cooling starts forming a new dome right here, and they can date that. So it was sweet. It was very romantic. So as they started doing some dating methods with it, it's very but as well played. But as they start, they actually discovered, like, okay, the method they used showed this thing to be way older than it was, which is ironic, especially given Zach's comment. So let's look at a picture here. This is a picture of Mount St. Helens Canyon, and so you can see the nice rock layers that were laid down. You can see the different patterns. From an evolutionary perspective, when you start laying down rock layers like that, you're looking at potentially thousands or millions of years. Okay, so they take a long time. It does not look like that was laid down over the course of weeks or months, but it was. And so these ash and rock layers were laid down. They had enough time to harden up just a little bit, and then when the water came ripping through, carved yourself a nice canyon. And so you got that laid out there right now. Okay. Now, the reason we make a big stink about this is really, it comes down to this. Without millions of years, evolution absolutely cannot work. Now, I'll make the case later that with millions of years, it still can't work. But if the Earth is young, there's no way. All right. There's just, it takes too long for these adaptations to compound. It takes too long for this stuff to happen. So let's let's talk about why they think that the Earth is old. They think the Earth is old based on what they call dating methods. So you've got radiocarbon dating, you've got carbon-14, you've got other methods. A few of them that you're going to use are things like the carbon-14 dating. Okay? I'm going to explain these so you can leave some space. You don't have to write all the names down. This is just an overview. There's going to be a lot more depth you can get to. But carbon-14 dating, uranium, lead, potassium, argon, these last two are going to be used to date things that are older. Carbon-14 is going to be used to date things that were once alive. So that's how you separate it. So carbon-14 basically functions, if you were alive at one point, they can use that to basically figure out how long you've been dead. Potassium, argon, uranium, lead, that's going to be used primarily on rocks and things like that. And it's going to be more or less trying to figure out when you were formed. Okay? Both of them operate on this principle that over time, you lose things. In the case of carbon-14, there's going to be certain elements that you lose. In the case of uranium and potassium, you're going to have one element changing into another element. And so as you compare the ratios of those two elements, you basically can make a statement and say, I can make an assumption on how old this rock is by what this ratio looks like. So if I were to be very brutal with my explanation, if I have a ratio that looks like this, and then I have a ratio that looks like that, if this is young, you can tell that this one is old. So like if this is one element and this is all that you find in the rock, and then suddenly you have only that much, you can kind of tell one rock is young and one rock is old. It's a very rough explanation, but that's...
Explain the chemistry behind it.
Oh, I will. All right. So that's the basics of how it's functioning. Other times, all they're doing is they're looking and they're saying, so here's your rock, and let's say I'm finding some helium in there. All right. So if I'm finding helium in there, it means this rock's probably young. If I think I should find helium, but I find none, it means it's probably old. That's another way they kind of figure this. So there are times where they're going to use a bunch of different dating methods because there might be a bunch of different elements in there. And so there's a whole lot of stuff going on. But let's start off with carbon-14. Carbon-14 dating functions on this idea. Now, if you know nothing about chemistry, which, let's face it, chemistry is one of those classes that we take and then we start to forget pretty quickly. Is that a fairly accurate assumption? Yes, okay. So now, in chemistry, you have the wonderful element of carbon. All right, carbon, it's number 6 on the periodic table because it has 6 protons. In the case of carbon, you're going to have 6 protons, and you're also going to have roughly 6 neutrons. So when you add those up, you get what? 12. All right. So your basic average, like almost every carbon atom you're going to run into is going to be what we call carbon-12. That make sense? Mm-hmm. Okay. The number of protons is what determines what element it is. The number of neutrons is going to help determine whether it's stable or not. Make sense? Mm-hmm. Okay. Now electrons play a role, and there's all sorts of wonderful things that go along with that, but that's more or less a summary of what we're dealing with. Now, as you add neutrons, okay, so protons equal the element. Neutrons are going to tell you what isotope it is. Because as I add neutrons, it becomes a slightly different substance, all right? So, for the case of carbon-12, if I add another neutron, I get carbon-13. Because now I still have 6 protons, and I have 7 neutrons. If I add another neutron, what do I get? Carbon-14. Now, this is where uniformitarianism comes in, because right now, in our atmosphere, we have a ratio of 1 trillion carbon-12 atoms for every 1 carbon-14 atom. So how this dating method works is entirely based on ratios. It's based on a ratio of 1 trillion to 1. Now, carbon-14 is constantly coming and going. Carbon-12 is changing. You've got a lot of different elements that play a role here. Sunspot cycles are going to have an influence on this. The amount of carbon in the atmosphere is going to have an influence on this. There's a lot of different things that actually can change some of these ratios if you really want to think about it. But in general, 1 trillion to 1 is our ratio. That makes sense so far? I know some of this stuff is like, what? You lost me at protons. Okay, but I'm trying to explain it as thoroughly as possible without losing you. But carbon dating is entirely based on this concept right here. Because carbon-14 eventually goes away, that's why you can kind of figure out, all right, how old is this object? All right, now how this works, let's just kind of go into this. This is what it would look like. So carbon-12, 99% of carbon atoms are going to be carbon-12. About 1% are going to be carbon-13, and less than 0.1% are going to be carbon-14. Okay, so that's how this is going to work. All right, so scientists, they can do, I don't know exactly the device they use. I don't know if it's a mass spectrometer or whatever, but they can use certain devices that basically will, it's not exactly like a Geiger counter, but you know that idea of where it will like vibrate so many times and you can kind of figure out, okay, that's how many we've got. And so I don't think they're doing this by hand, okay? And so you can kind of guess, not guess, you can kind of figure out what are these ratios. All right, now as you go, here are some of the problems that we run into. First of all, the rate of decay we have to assume is always the same. So if the rate of decay doesn't always stay the same, then we might have a problem. Okay? The ratio must always remain the same. If there's ever a time where the ratio is not the same, this whole dating method falls apart too. Because again, we are assuming, we know how long it takes for these things to decay. We're assuming that the ratio is always going to be 1 trillion to 1. If either of those things fall apart, then that's a problem. Now, these last two on this slide really shouldn't be on this slide, but they are. In radiometric dating, which is one element decaying into a different one, we assume that all the presence of those other elements are a product of decay. That's a problem assumption, but we'll assume that. And then one of the things we find in diamonds is we find that diamonds actually still have carbon in them. Specifically the type of carbon they should not have anymore. Because again, diamonds should have lost all of that long time ago. And so scientists, when they find carbon in diamonds, which should have decayed a long time ago, they assume that it's a product of that diamond being contaminated by an outside source. Now again, if you look at some of these things, you then say, all right, so if carbon can contaminate it from an outside source, or a daughter element can contaminate it from an outside source, how can we trust any of the material that we are studying to not have been contaminated by an outside source? Again, applying Occam's razor. If we have to say, ooh, this thing is younger than it's said to be because it has carbon in it or like it's not supposed to. Okay, it was just contaminated. Well, if you can contaminate a diamond with carbon, what else could you contaminate with carbon? All right, so let's take a step back and let me explain. Over time, carbon-12 and carbon-14, their ratio is going to change, and the average is about every 5,800 years. Okay, I'm going to double check that number just to make sure I've got. Because that is the number that I've taught this many times, and I keep, yeah. So about every 5,500 plus years, it goes away. All right, so carbon-12 to carbon-14, that ratio is going to be 1 trillion to 1. And after one period of time, which is about 5,500 years, you'll now find that ratio of carbon-12 to carbon-14 to be 2 trillion to 1. All right? Then it's going to go another cycle. It's going to drop again, and it's going to keep changing until basically until about 58,000 years. No more carbon-14 detectable. All right? So this right here is called a half-life. So the half-life is about 5,500, 5,800 years. So what's going to happen is every 5,800 years, that ratio is going to change. So right now, so let's say you die and we do a carbon dating test on your dead body. Okay? Because you're taking in carbon and losing it, taking in, losing it, all of that stuff, you will, in your body right now, have a ratio of 1 trillion to 1. The moment you die, you're no longer taking in carbon. Okay? So you'll start to lose the carbon-14 that's inside of you, but the rest of the carbon's pretty much staying where it is. That carbon-14 eventually is going to go away, and after the course of 5,500 years, if somehow your body got miraculously preserved, over the course of about 5,500 years, you now have 2 trillion carbon-12 atoms for every 1 carbon-14 atoms. And then 4 trillion, and then 8 trillion, and then 16 trillion, and then 32, okay? It's just going to keep halving because it's basically dropping like that.
Is carbon-12 more stable than 14?
Yeah, carbon-12 is the stable atom.
But 14 is not.
Yeah, it's going to decay. I believe it becomes nitrogen. So, okay. Who have I lost? Okay, so.
You haven't lost me, I'm just like, okay.
All right.
Very interesting.
So, that's why I'm not going to go much deeper. All right, so
I can go.
how it works is as you keep seeing this ratio change, you can figure out, again, assuming the decay rates have remained the same, assuming no carbon is corrupting it, you can assume it's been dead for that long. That make sense? Okay. After 58,000 years, so after about 60,000 years, if you want to round up, you will not be able to detect carbon-14 anymore. So carbon dating no longer works for that. Okay? So the maximum age you can date anything with carbon-14 dating is about 60,000 years.
It's the maximum age.
Maximum age.
You can't
So
date
just,
anything over 65.
okay? Does that make sense? All right. Now, like I said, these are the assumptions they're operating off of. They're assuming, which we don't know because we only have a small record of experiments, we're assuming the decay rate is always going to be the same. We're assuming the ratio of carbon-12 to carbon-14 is always going to be the same. When it comes to the next ones we're talking about, we're always assuming that the daughter elements are a product of decay. And then we also assume, so diamonds, they date diamonds to be way older than 58,000 years, but we find carbon-14 in them. So the question is, how on earth would that get in there? And the answer is, well, it was contaminated by an outside source. All right, so let's take a look at diamonds. All right, diamonds, like I said, you should not have any detectable carbon-14 after about 10 half-lives, like I said, or about 58,000 years. But again, it can be found in these things that absorb and release. So diamonds are, diamonds were once coal, which was once a living thing. So theoretically, diamonds could have carbon-14 in them, under a lot of pressure. You could become a diamond. That would be weird. My new experiment. People diamonds. All right?
Ooh.
So, now, if I have a normal rock, was that once alive?
No.
No.
Possible.
Can I use carbon dating on a normal rock?
No.
No. All right. So, back to my Mount St. Helens illustration. Mount St. Helens produced a lava dome after it erupted. That lava dome has various elements in it that will what we call radioactively decay. All right? And so it's things like uranium decaying into lead or potassium decaying into argon. The way radioactive decay works, sometimes it's a matter of protons being lost. Sometimes it's a matter of protons and neutrons being lost. There's different types of decay. All right, if you really, really care, there's like alpha, beta, gamma, there's a bunch of different radiation types. All radiation is one element decaying or becoming a different element, and it's shooting off protons and neutrons at you, or other things. And some of them are more harmful than others. All right? So if I am a rock, and let's say inside my rock I'm filled with uranium, over the course of time, uranium will start to shoot out radiation and eventually will become another element called lead. So uranium decays into lead. Chemical symbol for lead is Pb because of the old word plumbum, which is perhaps the greatest word ever. All right, you are plumbum. All right.
Jack is plumber.
I'm a plum.
Nope. So that is how it works. All right. In this illustration, uranium is what we call a mother, and plumbum is the daughter.
I'm a daughter now.
All right? That's how this works. So over the course of a half-life, which again is so many years, and each element has a different half-life, but over the course of time, uranium will decay and it'll become something, something, something. Eventually, it will decay into lead. When it reaches lead, it's now stable. Okay? So the mother is always unstable, and the daughter is always stable until she becomes a mother, and then she becomes unstable, and the daughter is stable. All right? And so that's how this is working. All right? So you've got an element that is not naturally stable. Okay? It's there. It's decaying and slightly insane. And over the course of time, it eventually becomes something else that is stable, no longer insane, no longer decaying. Okay? Does that process make sense? So, when a new rock is formed, according to this dating method, we are assuming that all the elements we find are mother elements, and that any daughter element we find was a product of decay. Does that make sense? So that's why when I drew this illustration, I said this could be a relatively young specimen, especially if this is all mother and this little part is daughter, because it hasn't been decaying for too terribly long. But once you get to the point where this is all the mother you have left and this is all daughter, now this is an old rock. Make sense?
You're an old rock.
Okay? Here's the problem with it. There are times where you do more than one dating method. So let's say you find uranium, you find potassium in there, and you find some other elements. If you do potassium argon and uranium lead and all those, you might get different dates, which is kind of weird. You think if you're dating the same rock, you should get the same dates. It just gets odd. But you do, sometimes you get the same dates, sometimes you get different dates, which again, just casts doubt on this process. But the problem is, there are times, so like when they dated Mount St. Helens.
Helen was a great mother.
I really wish there was a different word for this than dating. When you are,
Courting.
yeah, when you're courting these rocks, that's even worse. When you are doing the dating methods on these rocks, that's even worse. When you scientifically investigate the age of these rocks.
You're not
All right.
good at saying that.
So when they scientifically investigated the age of the Mount St. Helens rocks, what they found is these rocks were actually millions of years old.
What if the rock was?
But we know exactly when they were formed because they formed since the last eruption of Mount St. Helens, which took place in the 80s. Now, I don't know about you, but it sure doesn't strike me that the rocks would be millions of years old. But the reason is, to take the mother-daughter element illustration, they assume that the presence of all daughter element came from decay. That's not necessarily the case, though, because as magma and stuff pulls up from the earth, it may actually still have some daughter element when it forms that rock. Make sense? Okay. So again, radiometric dating. Some elements, such as uranium, undergo radioactive decay to produce other elements, By measuring the quantities of radioactive elements and the elements into which they decay, geologists can determine how much time has elapsed since the rock has cooled from an initially molten state. So that is the faulty assumption, because they are assuming that all the daughter element basically gets boiled off, if I can use that expression. That's the problem. All right, again, all age of the Earth dates, from what I've studied, come from extraterrestrial rocks. Now the reason this is because according to, observation and otherwise, most of the lower layers of rocks are constantly being basically burned down and reformed by the Earth. So that's mostly what happens. So as the Earth shifts and you got the plate tectonic drift and all that wonderful stuff, the lower layers, which would be older anyway, are getting destroyed and recirculated. So we don't actually have the oldest layers still in existence anyway. So that's why they date from extraterrestrial rocks, because you kind of have a pretty good feel for how old they are. So just a fun little note. All right, but so I'm talking like the age.
That aren't originally from the Earth.
So like meteors and things like that, they can do the dating. You can date an alien. All right. Now, like I said, there's some problems there. The assumptions, some of the bad assumptions, we've already kind of explained this. We're going with the idea that we know how much daughter element is present from the start and how much mother element is there from the start. So take this illustration. If you walk into this room and there's a candle burning, you don't know when I lit the candle. You could measure how long it takes for the candle to burn down, right? But if you didn't know how tall the candle was to begin with, could you have a fairly good guess on how long that candle's been burning? Ish, maybe. But here's some problems with that. So if I had a candle burning in this room, all right? So here's my candle.
Whoa.
The problem is, for one, I don't know when it was lit. All right? I don't know how tall it was. Okay? So I don't know either of those things, which has a really big impact on how long this candle's been burning. I can measure the rate of decay. But that doesn't tell me how long it's been burning, doesn't tell me how tall it was. All it tells me is this is how long it takes for this thing to decay. The other thing is, I don't know if there was some other thing involved here. So for instance, if I walk up to this candle, and you might have done this as a kid, and you start blowing on the candle just slightly, and so that it kind of gets that flame to melt a little bit differently, you may actually burn that candle down a little faster than it was supposed to. Or let's say I walked in and I blew the candle out for a while and then came back, and just before you came in the room, I relit it. And so I could say, oh, I lit that candle at 7 a.m. You'd be like, oh, okay, so this candle, by doing math backwards, that candle will be like 10 feet tall. No, it was actually off for a while, and I relit it like right before you got there. Those are some of those illustrations where it's like, we don't know what affects the decay rate. To our knowledge, it remains the same, but we don't know. We don't know how long it's been decaying. We don't know what daughter element was already there. They are assuming, again, based on uniformitarianism, they are assuming that all the decay rates have remained the same. All the daughter elements are a product of decay. They're assuming that nothing was there to begin with other than the mother element. That's a problem, and hopefully you see why. Like I said, there are times where you do it and more than one dating method works out and they all come to the same conclusion. But there are times, like with Mount St. Helens, where we're looking at a 20-year-old rock and it's telling us it's 1 million years old. Obviously that's flawed. Okay, questions on that? Because
Just for both potassium, argon, and lead.
pretty much all the radiometric dating methods, yeah. Okay, so there you go. The other assumption here is we know how much pollution got into that sample, which again, we don't know that. So just as with remember when we talked about carbon dating, and I said you have the diamond, all right, and the diamond actually has carbon-14 in it, and it shouldn't. That's a problem, because if the diamond is as old as they say it was, it should have lost that by now. And again, the problem is, well, if it could be contaminated, do we know how much contamination got in there? So, anyway, so that's just some of those things. I'm not saying the dating methods could never, ever, ever be trusted, but I'm saying we can't put our entire hopes on this thing because, again, there's some assumptions that just don't work. All right, so here's a picture from a museum somewhere. Multiple minerals from Mount St. Helens lava dome were radioisotope potassium argon dated by Steve Austin in June of 1992. Okay, so again, Mount St. Helens erupts in the 80s. Lava dome starts reforming. It's dated in 1992, so it's less than a decade old, if that. Geokern Labs in Cambridge, Massachusetts performed the analysis. The dates for the recent eruption of May 18th, okay, so it was there, was ranged from 300,000 to 2.2 million years. So it's quite a range. Given only 12 years from the eruption, there shouldn't have been any detectable argon present because it shouldn't have come about yet, all right? And so that's where, as you deal with some of these dating methods, they aren't as reliable as people like to make them out to be. Like I said, the other thing is different elements are going to give you different ages. If you pull a rock out of a certain layer and you don't tell them what layer it came from, there are times where lower layers are actually found to be younger than upper layers, which, if you think about that idea, how on earth would a rock layer buried underneath an undisturbed top layer be younger? But that sometimes happens. And so that's the thing. If we can't trust these for dates that we know, how can we trust them for ones that we don't? And so that's why I don't rapidly jump to, yeah, Earth is billions of years old, because some of the methods used to get those dates have been shown to be unreliable. All right. Again, uranium lead, it produces helium also as a byproduct. What's fun is helium should vent out and leave in about 14,000 years. But when we find helium in layers that should be older than that, what does that tell us? They're not older than that, all right? And so helium is one of those extremely light elements that should vent its way up through the rock layers and float away. Mount St. Helens, like I said, formed a lava dome. This is the one I was thinking of in 1986. When it was dated, the rocks gave a date of 0.35 million years old. So there's another one. There's an unpronounceable name, Mount Ngurahauhau in New Zealand, erupted in 1975. When they did dating methods on this, it showed that it was 0.27 to 3.5 million years old, which again, that's quite a span also. So like if I were trying to guess your guys' age, and I was like, Savannah, you're anywhere between 2 and 30. Nailed it. Yeah, I mean, that's not fair. All right, and so to be like 0.27 to 3.5 million years old, I guess in the grand scheme of like 3.5 billion year old Earth, that's actually fairly close, but it's still quite a span. All right, and so those are just some of the things there. Like I said, there's some problems with this. As we move beyond the dating methods, let me show you a few more areas that are a problem for evolutionists. We're going to dig into these a little deeper, but before I move on, any questions on the dating methods? I'll show you that yet, because I already did. This is Ernst Mayr's What Evolution Is. They believe that one of the teachings of evolution is this. So I'm going to read from you from What Evolution Is by Ernst Mayr. This book was written in 2000, I believe, and says, perceptive anatomists observed in the 18th century that the embryos of related kinds of animals are often far more similar to each other than their adult forms. Okay, we're not going to go into all the scientific terms for all this, but the idea is they thought that embryos, that the word is recapitulated, I had to use the term because I couldn't think of another word for it, but they basically rehearsed the evolutionary progress that they had made, and then eventually they diversify and become adults, or babies, I guess, and because they have to be babies first. All right, so The embryos of related kinds of animals are often far similar to each other than their adult forms. An early human embryo, for instance, is very similar not only to the embryos of other mammals, like a dog, a cow, and a mouse, but in its early stages, even to those of reptiles, amphibians, and fishes. The older the embryo, the more it shows the special characters of the higher taxons to which it belongs. Okay, so if you kind of think, all right, this is the animal kingdom, this is a specific group, this is the phylum that we belong to, those will all have similar embryo shapes and designs. That's what this is trying to say. So as you get older and older, like, you bear more and more resemblance to the things you evolved from, or things you had a common ancestor with. Okay? Now this is an evolutionary book. This is a book trying to teach what evolution is, and this is what it's saying. And this is it's published in 2000. Okay? So I'm laying a groundwork from the evolutionist's own mouth to say, this is what they're teaching, and I'm going to show you the diagram, and then I'm going to kick the stool out from underneath him and show you the wonderful science it's based on. All right, it says this: the older the embryo, the more it shows the special characters of a higher taxon to which it belongs. When the adults are highly specialized, for instance, the sessile barnacles among the crustaceans, their free-swimming larvae are still very similar to those of other crustaceans. Some of Darwin's opponents asserted that such larval similarities would prove nothing. All development by necessity, I can't read today, moves from simple to complex, they said, and the early developmental stages, being simpler, are thus more similar than the later, more complex ones. So the illustration we would say is, no matter what building you're building, the foundations are all going to look similar. The joists are all going to look similar. The studs are all going to look similar. Like, the basic building blocks are going to be there, and if they're performing a similar function, they're going to look similar. That's an illustration we would use. It says, this is in part true, but embryos and larvae always have some characteristics peculiar to the phyletic lineage to which they belong. So, if I am in a specific phylum, okay, let me take a step, make sure you guys are all on the same page with me science-wise.
Kingdom.
Okay, so we start off with kingdom. Now, it's gotten even broader nowadays, but this is what I learned, and I don't feel like relearning all of the other ones, because now they have like one above kingdom, and they have superphylums and subphylums and whatever. I'm just going to go with the basics. All right, so you got kingdom, phylum, class, order, family, genus, species. All right, so if I say I am part of the animal kingdom, all right, a good example of the phylum would be something like all right. You see how that, so kingdom would be animals, the phylum, or the phyletic inheritance that they are going to have, phyletic, okay, phylum, arthropoda. So all arthropods are going to share similarities. So what he's trying to say is that as you develop, you will share similarities with your phyla. So we are part of phylum Vertebrata. At least we used to be. Now it's Chordata or something. That's the process, all right? And so even if you get all the way down to a species, which is, like the common bumblebee or something, it's going to share similarities with the spider and the scorpion and all these different things, because they all come all the way back up here to arthropoda, even though they're different classes, et cetera, et cetera. Make sense? Okay, I'm assuming you had some rough biology at some point previous to this point. If not, there's your crash course. I think it says this. This is in part true, but embryos and larvae always have some characteristics peculiar to the phyletic inheritance to which they belong, thus reveal their relationship. So that's how this chart works. Furthermore, study of embryonic stages very often show a common ancestral stage gradually diverges in different branches of the ancestral tree. This leads to a far better understanding of evolutionary pathways. Okay, as he continues, he shows this diagram. This diagram is an extremely famous diagram dating from the 1800s, and you had a guy by the name of Haeckel. Haeckel made drawings which are known as Haeckel's embryo drawings, because they're drawings made by Haeckel of embryos, and they are drawings. So as you look at it, this is probably a chart that if you've done any looking into evolution, you've probably seen this chart. Have any of you seen this chart before? Okay. If you've ever done anything with Rebecca or probably Bob Jones, I'm not sure, they have probably had this embryo drawing in their book. Now, look at the caption underneath this drawing. Okay, so underneath this drawing, again, this is the drawings that Haeckel made. It says this, figure 2.8, Haeckel's figure of 1870. So it's pretty old. Showing similarity of the development of human embryos to 3 comparable stages in 7 other kind of vertebrates. So you got humans, rabbits, cows, pigs, chickens, tortoises, salamanders, and fish. And so they've got stage 1, or at least what he calls stage 1. You look across all those and you go, wow, they're similar. You look at all the stage 2s, and other than the fish and the salamander, which are older, you can look and go, wow, they're all pretty similar too. And then you get to the more developed stage, and you're like, wow. I mean, they're showing some similarities, but they're different because they're diverging at that point. That's amazing. So it seems to back up what he said, but the diagram has a caption. Haeckel's figures of 1870 showing the similarities of development of human embryos and 3 comparable stages in 7 other kinds of vertebrates. Haeckel had fraudulently substituted dog embryos for human ones. So he's claiming that these weren't human embryos, they were dog embryos, so that was the fraudulent part. But they were so similar to humans that these, if available, would have made the same point. This is an evolutionary textbook, not textbook, but evolutionary book, trying to prove what evolution is, written by an evolutionary biologist who is credited as really knowing his stuff. And that's what he says about Haeckel's embryo drawings. It was fraudulent in that he substituted dog embryos for human ones. Okay. Another book written about the same time is this book called Icons of Evolution. Okay? Written by a guy by the name of Jonathan Wells. And I want you to observe, because what he does is he actually goes all the way back and he shows what do these things actually look like and how do they actually look at the same stages. So look at that actual chart. Okay? So in the earliest stages, yeah, most things look alike because they're just a ball of cells. So I think we could all agree there. But look at this. When you start to have the cells divide, that's a bit different. I mean, some it's just this little some of it's a bundle, some of it's just creases. You know, look like a little alien egg sac about to hatch there. All right? And now you get some very rapid differences. Like they start piling up real quick. These do not look the same. These don't look the same. These, yeah, that's not exactly similar either. And so as you go through, this book is going to start digging into more and more, gives you a picture of what Haeckel drew and what they actually look like. So when we say that Haeckel, fraudulently substituted dogs in for humans, no. What he did is, this is what he drew. This is what they actually look like at that stage. So he didn't just fraudulently substitute one animal in for a human. He made up the look of the things altogether. Like, that's what they look like. So when we talk about Haeckel's embryo drawings, and I remember talking to my grandfather, or my mom was talking, and she passed the story on to me, he actually used that as an illustration of why evolution was true. So even back in the early 2000s, when that discussion was going on, and this has been known for a long, long time. This has not been something that was just recently discovered. Like, they have photographs that you can look at and show that these things are fraudulent. But Haeckel's embryos are one of those, what we call zombie things, because they just keep coming back to life. And they keep getting reproduced in science textbooks to today to prove evolution. That's just one illustration of where you can say, it's a fraud. Like, it's not true. And this is a book, both of these books were written about the same time, and yet this one, Defending Evolution, will straight up tell you, the only thing that's fake about it is we substituted dogs for humans. This one says, no, like the entire drawing is fake. So again, you can see why you would be a little skeptical when it comes to some of this stuff. Let's take a look at it. Again, it was late 1800s, Ernst Haeckel is trying to back up an idea called embryonic recapitulation, which is the big technical term for what I just described. That embryos go through evolutionary stages one more time. All right? Embryonic recapitulation. Embryos replay the evolutionary story in their development. It was a belief back then. It's not true, but it happens. To do so, he fraudulently modified and swapped different embryos in order to make it look as if animal embryos go through evolutionary stages. Just faked. Just completely made up. All right? So that's why, again, you'll understand why I'm a little skeptical when it comes to some of these things. His theory, I'm going to give you his phrase, or the phrase that they use. Ontogeny recapitulates philogeny, which is basically your development recapitulates your evolutionary history. You don't need to know that term, all right? But his theory was that your development in the womb replays your evolutionary history. It's not true, but that's what they claimed. Though it was disproven soon after, so remember, this is back in the 1800s, late 1800s, the images he forged are still found in textbooks and scientific literature to today. Again, I showed you a book from the 2000s. They just claim, he fraudulently swapped dogs with humans. No, he fraudulently drew entirely new embryos. All right? According to a lady who runs a group called the National Center for Science Education, the reason they use it is because they're free. And they're good images for the most part, so why not use them? No, they're entirely made up images. Like, you saw the drawings. That doesn't work, all right? And so that's another example of things that people keep using to try to prop up evolution that is not true, okay? Now, I will tell you, there are certain evidences people will bring that are actually good for evolution to use. Like, oh, okay, that's actually helpful. Like, that makes sense. If we have time, I'll show you some of them. Again, none of them discredit creation. But there are a few things where you're like, okay, evolution made a scientific hypothesis and it came true. That's cool. You actually have some things that actually support your cause. But what they do is a lot of times they're using these type of illustrations to be like, this proves it. And it's like, no, it doesn't. In fact, it's a lie. So there you go. All right, any questions so far? You got a few more illustrations and then we're done for this block of time. I want you guys to understand this. I want you to be able to potentially reproduce it. But what questions are there?
Are we fraudulently making pictures now?
Mm-hmm.
Awesome.
Okay. Another one here that we run into are fruit flies. Mutations. We've covered this a little bit yesterday, but let me give you some text on it. Mutations are copying mistakes that build up over time. All right? Like I said, when you look at DNA, you have the DNA sequence. Every now and then it copies wrong. Okay? Again, how much science have you guys had? A lot, little, moderate amount. Okay. So if there are terms I start using that you're like, I don't know this, just say it. But I'm assuming you guys have at least the basic understanding of like, there's this thing called DNA made of letters and the letters copy through RNA. There's some of those things. So as mutations copy, there are some of these mistakes that crop up, okay? So through mutations, you can sometimes get like a leg that comes out of an antenna of a fruit fly or something. It's really weird. It's basically copying stuff in either the wrong place, or you copy it twice, or you drop out a letter, or you add an extra letter. Those copying mistakes basically tell you, you need to build this here. And sometimes you're building it in the wrong place. So every now and then you get a fruit fly, like I said, with an extra set of wings, or an antenna that is actually a leg, or just something weird that it's a copying mistake and it plopped this thing in the wrong spot. I was listening to, like I said, that evolutionist, and one of his excuses for why evolution works is because you see this type of stuff happen and it's like, oh, there's a leg coming off this antenna. I don't see how that's helpful. If I had a foot coming off my face, that's pretty cool. It's a new development, but it's not helping me survive. And what you find with a lot of these mutations is they actually make the creature unable to reproduce, either because it sterilizes the creature altogether, or nobody is going to be like, yeah, those are the genes I want. I mean genes as in G-E-N-E-S, not J-E-A-N-S. But nobody's going to like randomly go up to something that can't fly. You know, if you're a fruit fly, you know, and you're looking for a good date, you're not going to look and be like, oh, I want him to be the father of my babies because, you know, he has 4 wings instead of 2, and he can't fly with any of them. That's where natural selection comes in to say, you ain't getting picked. So yes, you have a mutation that gave you an extra set of wings that could be really cool, but you're not reproducing. So therefore, that mutation dies with you. All right? So that's the thing. Mutations pretty much incapacitate you, or they're going to be a loss of information. There are several types of mutations. First of all, there's a substitution where a part is swapped out. Okay? When we talk about parts being swapped out, like I said, it is the information should have read this way, but because of a glitch, it read this way. And again, you're simply combining letters. It's like if I wrote a sentence, you know, the big gray dog jumped over the fence. All right? And as I write this, and let's say I'm like, okay, we're going to copy this. So you come along, you insert your copying mechanism, and then you start replicating it, and accidentally I trip a little bit, and I'm like, and I accidentally spell gray that way instead of that way. Okay, that doesn't make a difference. I'm just substituting out a letter. It probably is going to produce the same effect. But if I trip and I go, and I swapped out the D in dog for an H, now it says the big gray hog jumped over the fence. That gives you a completely different picture. But all I'm doing is I'm just substituting one letter. So this happens occasionally in genetics as things are being copied where something messes up. And so now your offspring is born and it has an extra iris or something. And you're like, huh, that's cool. I knew a guy who actually had 2 irises in his eye. It's kind of neat. Some of you know him. But it just kind of had like double, like snake eye. It just, I don't know how that works, but it's just a mutation. It just happens. All right? Again, they're not always lethal. They're not always harmful. But in general, they are. All right? So that is substitution. The next one is what we call insertion. Insertion basically works in that a part is plopped in where it shouldn't be. And so it would be, you know, the big gray hog hopped over, jumped over. Yeah, that's a jumped. Jumped over the fence, and then you insert fences. It's just inserting a new letter, all right? But since these things are read in sets of 3, every now and then when you insert a letter, it bumps everything else down, which actually could create a cascading effect of, ooh, I now have a whole new set of proteins being put together because it reads it by 3. So if I have 3, 3, 3, so on and so forth, you see how this ending, if there was more to this sentence, could actually mess up everything else. Now again, typically that's going to kill it. And there are actually mechanisms in to kind of keep an eye on those type of things. But again, it's insertion. You also have deletion. And so, again, it could be the big gray jumped over and you're like, the big gray what? Now again, that's going to mess up your whole sentence if it fell there. But if it fell over here, where the big gray dog jumped over fence, you can still make sense of that. You can still move on and manage. So it depends on which part gets deleted, whether it was something that was, I don't want to say redundant because it's not redundant, but there are certain genes that are not active at the time. And so if you drop those off, they're not a huge deal. In fact, there are times where you drop a gene off, it helps you survive better. I talked about that a little bit yesterday with some of those bacterial experiments. And so that's the idea of a deletion. But also you have inversion. And so this is where it could get really weird. Where it was, the Gib gray dog jumped over fence. And you're like, are you okay? You see, over time, this becomes more and more nonsensical, the more and more, and eventually it could become a completely different sentence, especially if I did it like this. Let's say dog got inverted. Now I'm stepping into divine council territory or something. You see, like, I'm swapping, and so now this sentence completely changes meanings because certain ones got swapped. Now, in DNA, it's a little more complicated than this, because those DNA molecules are a little more, when you have 4 letters to make everything, those letters are a little more important. So, and it takes more than just 3 letters to make a protein. So when you start combining, basically it would be like, here's my paragraph to make this protein. Okay, so there you go. All right, almost 100% harmful, always result in a loss of useful information. But again, it doesn't mean you're less likely to survive, because there are times where certain mutations drop parts off. So there have been things that have, there have been investigations into, for instance, dogs, where there's certain genetic traits that will make them less aggressive, but it also takes away certain other survival characteristics.
Like Zach's dog.
So there you go. Any questions on that? That's another thing that evolution has to go to. It has to go to mutations and natural selection. And the problem is, you don't always detect that there's a bad mutation right away, as we kind of explained yesterday. So I'm hoping you remember some of what we said yesterday, some of what you learned elsewhere, and you put them all together and you get kind of this picture. Here's some pictures of some fruit flies, just to kind of give you an idea of the different ways fruit flies can mutate. You can get different eyes, you can get different wings, different other parts. Sometimes you can get a duplicated thorax or whatever. It happens. Those are all mutations that are possible. Now, these poor things are getting like baked in radiation and all sorts of stuff. But sometimes you just leave them alone and you do it, and random things pop up. But you'll notice there's nothing that will specifically help it survive so much better that it will grow on to become something completely new. In fact, when you do see things like an extra set of wings, typically it doesn't have all the other structures needed to make those wings work, so therefore it would never survive in the wild anyway. So yes, good mutation, you got extra wings, that could be really helpful, but it's going to take a little bit more work to make that work, which it won't have time to do. This one is fun though, and this like I said, is where we're going to end. They have found red blood cells in dinosaur fossils. Just to kind of put it in perspective, you should not still find those type of structures in dinosaur bones, because they should have rotted and basically disassembled themselves millions of years earlier. Like, there is no way it should have preserved that long. Here's the thing. In a supposedly 65 million year old T-Rex, they found visible, like they could detect blood cell tissue. Again, that rots really fast. Should not work. I'll explain some of how they excused it, but I want you to see how this worked. It's claimed that iron released from the hemoglobin may have preserved it for longer than we otherwise thought it could. If you look at the experiment you did, you can see why they would come to that conclusion, but I also want you to kind of see how far your unbelief can stretch. In short, what they did is, I believe they took a goose, like the bones of a goose or a turkey, some bird, and they infused it with iron, and they found out that when you infuse it with iron, it will preserve it 200 times more than it should. So you extrapolate from there and you say, all right, so if we have, dinosaur bones, for instance, a 200-fold extrapolation would bring you out to, the 65 million years old. So that's where they're going. So they're saying, if you have a bird and you can make it last 200 times longer than it should, then theoretically, if you do the same thing to a dinosaur bone, it should last 200 times longer than it should, which, solves the problem. All right, so that's their method. So that's the 200-fold increase in stability. Again, I ask you, we're not scientists in this room. I get that. We're not like brilliant minds who, you know, do all this stuff. But applying Occam's razor, which is, let's explain this in a way that doesn't require all sorts of random stuff to make up. What makes more sense? The fact that this dinosaur bone has not been buried for 65 million years, or, well, somehow a massive infusion of iron got put into this dinosaur bone. I mean, it must have had just a ton of blood that just all leached the iron in there. And so it somehow has preserved it for 65 million years. So again, you look at some of these things and you kind of say, this seems to be evidence. Like, we can't 100% prove it, but this seems to be evidence that this dinosaur lived more recently. And it seems like you're stretching. You're reaching for variables to be like, well, we got turkey bones or goose bones to last 200 times longer, which in that case is like a matter of months, you know, or years. I mean, we're not talking decades. We're not even talking centuries. We're talking a 200-fold increase where it's like, well, it would have rotted after a day, but it lasted a month. You know, like those are like some of the ways they go about this. All right. So again, like I said, the iron preservation theory. So does that work? So to go from, you know, the blood vessels in my bird bone lasted 200 times longer, so therefore I think that blood vessels in a animal bone could last, and really it's a 99 million fold increase if you think about it. How does that work? So anyway, hopefully you understood the basics of that. But you also still need rapid burial. You also need the infusion of iron to do that. You need a lot of other stuff. And that's where, as we study out evolution, there's a lot of those things where it's like, yes, you may have explained this one part, but what about all these other things that have to happen simultaneously? So typically when something dies, it doesn't get rapidly buried and then pressed into a rock and preserved that way. Most things, when they die, they get rapidly eaten, or they rot on the surface of the earth. And then, you know, but for a dinosaur bone to fossilize, this thing is dying, is quickly being buried so it can't get eaten first, and then the iron somewhere in that whole mix has to get in there, and the iron is going from what they found to be a 200-fold increase to almost 100 million-fold increase. Again, the theory is there, but I don't think that really passes what we call the sniff test. So anyway, we'll watch that when we come back. But I want you guys to see kind of how this stuff works. All right.